Hello, in this video we will see receptor tyrosine kinase. After a quick introduction, we will see pathways involved in signaling downstream of these receptors. Let's get started. In the video on receptors, we have seen that receptors are molecules that first receives the signal from the external messenger. Catalytic receptors are receptors with inbuilt enzymatic activity. Receptor tyrosine kinase or RTKs are such catalytic receptors. Most receptor tyrosine kinases are single-pass transmembrane proteins. They have a ligand-binding domain on the extracellular side and tyrosine kinase domain on the intracellular side. Speaking about the tyrosine, these are some tyrosine residues among many amino acids in their structure. Examples of such receptors are receptors for insulin and various growth factors like epidermal growth factor, platelet-derived growth factor, etc. So this was the basic about receptor tyrosine kinase. Now let's see the signaling pathways involving these receptors. Under resting state, these receptors are in monomeric form. The binding of the signal molecule causes two such receptors to come together and form a dimer. Now the kinase domain of each receptor adds a phosphate group to tyrosine residues on each other. This results in the formation of phosphotyrosine motifs. They serve as a high affinity binding sites for proteins with SH2 domains. SH2 domain is basically a part of a protein that can recognize and bind phosphotyrosine. It is seen in many intracellular signaling molecules. So downstream effect depends on which exact protein is recruited. There are two commonly seen pathways. One is MAPK pathway and second is PI3K pathway. Let's first see MAPK pathway. As usual, binding of a ligand with the receptor causes dimerization and then transphosphorylation of tyrosine residues. Now, GRB2, which is an SH2 containing protein, recognizes phosphotyrosine residues on the activated receptor. GRB2 then attracts son of 7-less or in short SOS. Next we have RAS, a type of small g protein. SOS catalyzes the removal of GDP and its replacement with GTP. This activates the RAS. The RAS then activates RAF1, which is also called MAP kinase. RAF1 phosphorylates and activates MAC, which is also called MAP kinase. Then MAC phosphorylates and activates MAP kinase, or in short MAPK. These confusing names are actually convenient. This last one is MAP kinase. This one phosphorylates MAP kinase, so it is called MAP kinase. And this one phosphorylates MAP kinase, so it is called MAP kinase. These three guys typically assemble on a scaffold protein. It brings them closer for interaction and thereby speeds up the activation process. Now MAP kinase phosphorylates various proteins involved in cytoskeletal assembly and cell matrix interaction. This leads to changes in cell morphology and cell migration. MAP kinase also goes to the nucleus and phosphorylates various transcription factors. This leads to an increase or decrease in transcription of genes involved in cell survival and proliferation. If you are getting confused between so many molecules, forget the detail. Just think receptor and all the second messengers are lined up like dominoes. Ligand activates the receptor, leading to series of activation of downstream molecules one after another. It's like a series of dominoes. And at the end, we have the final cellular response. I hope this helps. Now let's talk about phosphatidylinositol 3 kinase or PI3K pathway. Again, activation of the receptor causes the formation of phosphotyrosine motifs. This time they recruit PI3K, an SH2 domain containing enzyme. Its substrate is PIP2 in the membrane. PI3K phosphorylates PIP2 to produce PIP3. PIP3 serves as binding site for many proteins like 3-phosphoinositide dependent kinase 1 or PKD1, PKC, and guanine nucleotide exchange factor WOW. Let's move ahead with PKD1. It is seen downstream of the insulin receptors. As it binds to the PIP3, its kinase activity leads to phosphorylation and activation of AKT. AKT is also known as protein kinase B. AKT in turn regulates multiple target proteins. For example, it inhibits glycogen synthase kinase 3 beta. So this was PI3K pathway. Now let's have a quick summary. Most of the receptor tyrosine kinases are single-pass transmembrane proteins. They have extracellular ligand-binding site and intracellular tyrosine kinase domain. Receptors for insulin and some growth factors are receptor tyrosine kinases. Under an inactive state, receptors are in monomeric form. Ligand binding causes dimerization. Some receptors, like those for insulin, are present as dimer even under resting condition. In the dimer, tyrosine kinase domains of each receptor phosphorylates tyrosine residues on each other. Resultant phosphotyrosine motifs attract SH2 domain containing proteins. In MAP kinase pathway, downstream signal molecules are GRB2, SOS, RAS, RAF1, MAC, MAP kinase, proteins involved in cytoskeleton assembly, and transcription factors. PI3K pathway involves PI3K, PIP3, then PKD1, PKC or guanine nucleotide exchange factor WOW, and downstream to PKD1 we have AKT and glycogen synthase kinase 3 beta. That's it for this video. If you feel this video will help your friends and colleagues, please share it with them too. And don't forget to subscribe because lots more to come. At Nonstop Neuron, learning medical concepts is as easy as watching cartoons. Thanks for watching. See you in the next video.